So, wow, thank you everyone for coming tonight. That's quite impressive with the Dodgers playing in the fourth game tonight. So I'm actually rooting for the Yankees. I was just discussing. I know because it shouldn't be 4-out. I want the Dodgers to win, but we have to throw them like something. So we don't have to. No.
This is being recorded.
Oh no.
Detroit,
but not the Yankees. Fair enough. Well, we'll see what happens because I don't know. It won't be long now. Thanks anyway for coming out tonight for the community lecture series in the middle of all these events. It's great to have people come from, I can see across campus, but also from the public to participate. It is my great pleasure to introduce this evening Dr. Christopher Hughes, who is a longstanding member of the Stem Cell Research Center, and really, I think, illustrates some of the interdisciplinary elements that are so important to what we do here. So Chris was the longstanding chair, we were just talking about it a minute ago, of molecular biology and biochemistry. He's chancellor's professor of molecular biology and biochemistry on campus here. He also was the director of the Edwards Life Sciences Center for Advanced Technologies and Cardiovascular Research, among his many aspects of service to the campus overall. And his research is really interesting in terms of understanding cell biology and how we need to harness new technologies to ask better questions, right? So if you think about stem cells or culturing cells, like putting cells on a dish, which is a lot of where we started in stem cell biology, we take cells out of their natural three-dimensional culture environment, the body, right, in an organ or a tissue. We put them on a flat, hard surface and we ask them to behave normally. And that is not something that cells can do well. So at the interdisciplinary face of moving things forward for cell and tissue research, we need to ask better questions. We need to put them in contact with cells that they would see normally in an organ or a tissue, right? They're partners in crime. And we also need, it turns out, to show them something even more important, which is a structure or a surface that is pliable, that isn't just hard, and that is vascularized, that gets flow. And that's because so much of development in our bodies is really dependent upon establishing blood flow, establishing a vascular system, and that changes the pressure of the tissues that those vessels sit in all the time, right? It's causing an expansion and contraction with flow, and it's causing just flow through those structures. So the molecules that are delivered that are different, the mechanical cues are different, and having multiple cell types around that all talk to one another is different. And this is where our field has been moving for some time. And this is the space that Chris works in. He's made incredible advances there. And he also is really interesting because he's an example of how biotech and an academic setting communicate, because as you'll see in his disclosures, he's taken the step of bringing what he does in the lab to make it available more broadly by launching a biotech company. And so with all of that incredible expertise and that interdisciplinary research, please welcome with me Chris Hughes tonight, and we'll hear his talk. We'll have questions at the end, plenty of time for questions, so don't worry.
Thank you.
Thank you.
It's going to go a lot quicker than you thought. I can skip my first 10 slides. All right, so I was told that as this is a community lecture, I should sort of pitch this for the general public. I see several people in here who are hardcore researchers, so apologies to you if some of this gets a little bit simple, but I think it's good to go through it. So yes, disclosures. I have to put this up, and I have to tell you that some of the technology I'm going to show you has been commercialized through this company. I founded it, I'm a shareholder in it. But rest assured, everything I do is watched very closely by the Conflict of Interest Committee, and they are so far happy with the way things are going. So good. So many years ago, I came home from work, long day, come in, and my daughter's there, and she goes, "Hey, Dada, how was your day? Did you cure anyone?" I'm like, "Well, honey, it doesn't really work that way. I'm a basic scientist, and what we do, we try to understand the basic mechanisms of how cells and tissues work. And I can see she's already drifting off. And I say, but eventually, the things we do, people will make drugs and they'll get to the clinic and then they'll help. Oh, where did she go? So at that point, I thought, I think I would like to be a little more at the pointy end of things, do some translational work and get closer to the patients. Maybe actually one day be able to say, oh, I did actually. So that's where my sort of shift from pure basic research to more translational arena started. So what I'm going to tell you about today are a couple of diseases we've worked on. One is called port-wine birthmark or Sturge-Weber syndrome. The other is hereditary hemorrhagic telangiectasia. And for fun, I could point to somebody and ask them to say it, but it's hard. It took me a while to figure out how to say telangiectasia. So we just call them PWB and HHT, which is much simpler. So from now on, it'll be PWB and HHT. All right, so the problem with these diseases is that blood vessels don't form properly. So that's the core issue that we're dealing with. And I'm a vascular biologist. My PhD was in vascular biology. I've worked in it since a long time ago. Can't count how many years. So at core, I'm really into blood vessels. That's what I do. And so I particularly wanted to work on these two diseases because they are problems with blood vessel development. So let's talk about blood vessels. So a lot of people tend to think of blood vessels as just being plumbing, right? A bunch of tubes carry blood around the body, and to some extent they are, but they are living and they are dynamic. They can get bigger and smaller. They can make new vessels. They can regress and take away vessels that aren't needed. They control what crosses from the blood into the tissue. They control what goes from the tissue into the blood. They carry immune cells around. They tell immune cells whether this is the tissue they should be going into or not. So in all these ways, they're kind of interesting. They're dynamic. So some early discoveries in blood vessels. So Galen was a Greek living in Rome. I'm not sure he wanted to be there, but he was. And he first proposed that the heart pumps blood out through the vessels to the periphery. And bizarrely to us now, that was good enough for him. The heart made the blood. It went to the periphery and I don't know, just disappeared and just got sucked up by the body or something. So the idea of circulation didn't come in until Harvey, which was, can't even do the math, 500 plus years later. He suggested that blood actually recirculates, comes back to the heart, so a closed system, but he wasn't really sure how. So you could see big vessels going to tissues and big vessels coming back, but nothing in between. And it took the invention of the microscope by Van Leeuwenhoek for Marcello Malpighi to discover these very tiny vessels called the capillaries, or capillaries, as we have it over here. I will probably say capillaries because that's what I grew up saying. All right, so you can think of the vascular system kind of like the highway system, right? So here's Southern California, and you've got your big roads, the 405, and you'll often hear traffic reports talking about the major arteries into the city are blocked, right? So we've even used that terminology. So there are big vessels in your body, one coming from the heart, called the aorta. That's kind of like the 405 or the 5, though hopefully flowing a little better than those roads do. And then as you come down, you'll start to get smaller roads come off, like the 73 and the 55, right? Those are the branches. You can think of those as the smaller arteries coming off. And then you get down to, okay, you can't really see that very well, but you need a microscope. So in here somewhere are the small roads like Jamboree and MacArthur, and then those lead very smallest roads to your house, right? Every house is on a road, pretty much. And then every cell in your body is next to a blood vessel. So while you get deliveries to your house, Amazon drops stuff off, trash guys come, take stuff away, blood vessels do the same. They deliver nutrients to the cells, and then they take away the waste. So in that sense, you can think of your vascular system like a highway system. And there are a lot of blood vessels in your body. Vascular biologists tend to think of your body as being a bunch of blood vessels, yeah, with some other cells around. And turns out that every cell in your body is within 100 microns of a blood vessel. So when you look at the sizes of these, here's your aorta, comes off the top of your heart. You could, don't recommend it, stick your thumb in it. That's about the diameter of the aorta. And that goes down to the smaller arteries and down to these very small vessels, the capillaries. And they're about 1/10 the width of a human hair. So think of a hair, think of 1/10 of that. The smallest capillaries are that size. And then you go back to bigger vessels to complete the circulation. So this is how your vascular system works. You have your arterioles. They carry oxygenated blood from the heart and the lungs, carries oxygen out to the tissues, gets the tissues through the capillaries. And then that deoxygenated blood goes back through the veins, back to the heart and the lungs, round and round you go. And so this is what a blood vessel looks like. And it's lined by specialized cells called endothelial cells. And we're going to be talking a lot about those. So here are your red blood cells. And so this is a little capillary with the endothelial cells. So that's the structure of blood vessels. And a little taste of what's to come. We found ways to create blood vessels in the laboratory. So we can grow them in a dish. And here's one. And the arrows here are pointing to the outside where the endothelial cells are. This little star is pointing to what we call the lumen. That's the bit down the middle. So the blood vessels are a tube, right? So they're hollow, and that's the hollow part there. And this is just a prettier version of that. What's different about this blood vessel and this one is that these ones don't have blood flowing through them. They're empty. So not optimal, but we learned a lot from making blood vessels like this, as you'll see. All right, little diversion here because, hey, who doesn't like dinosaurs, right? So this is your cardiovascular system, and you know your blood pressure, like 120/60, right? So 120 is measured here, your arm, that's at your heart. So that's your blood pressure. And because if I'm standing up, gravity is pulling all my fluids down. And so the pressure in my feet is higher and it's lower in my head. So remember as a kid, you did a headstand and like, oh, all the blood rushes to your head. Well, it kind of does. And the pressure goes up and poof, feels like your head's going to explode. So that's that. And you're already familiar with this idea of pressure lower down. If you go to New York or big cities, you'll see these water tanks on the roof. And notice how the ribs get closer together as you go down. That's because the pressure at the bottom is higher, just as it is in the body. So, yes, you did hear me say dinosaurs. So this fella here, how we used to draw our brontosauruses reaching up to get things out of the trees. And if this fellow wants to keep 90 millimeters of mercury pressure in his head, same as we have, he'd have to have 700 at the level of the heart. Now, to generate that kind of pressure, his heart would have to weigh 5 tons. Now, these guys were big, but they weren't that big. And that's one of the reasons that we now think that this long neck was actually so they could reach out over a swamp. Because if this guy actually put his head up like that, he'd faint. So there'd be no blood going to his head and he'd keel over. And obviously not a great idea, so we think he probably kept his head down most of the time. Anyway, bit of a diversion, kind of fun. So back to the story.
No, that was good enough. We're thanking.
Yeah, good. All right. The takeaway message.
Don't stand up suddenly.
Don't stand up suddenly. I get that. If I stand up too quick, get lightheaded. You imagine if your head's way up there. All right. So we want to understand diseases. We want to understand vascular diseases. And so what we do as biologists, we create models of the disease. it's hard to work on people, right? Doing experiments on people is called a clinical trial. It takes a long time, it's expensive, it has risks. So we try and create models of diseases. And as somebody famously said, I blanked on his name, all models are wrong, but some are useful. So we try and make the best models we can and learn from them what we can, but understand that they are not perfect. So, for example, we could do experiments in mice, and lots of scientists use lots of mice. So the question is a person equivalent to a mouse? Now, I would ask a lay audience, and they would laugh and go, well, of course not, right? We're 2,500 times bigger than a mouse. We live 50 times longer. Your hearts right now are beating, hopefully, somewhere between 65 and 70 beats per minute. A resting mouse, 300 beats per minute. If you get a mouse excited, that heart's beating 700 beats a minute. It's vibrating. So is that a good model for a person? Probably not. That's not to say that mice haven't been useful and aren't useful. We've learned a huge amount. For some things, they're great. For some things, they're not. So what do we do? So a little history, my journey towards making blood vessels. So how do we get to where we are now? So this is my lab when I first started here in 1996. That young fella up there with the brown hair, that's actually me, believe it or not. And so what we did, we took endothelial cells from blood vessels and we grew them on dishes, plastic dishes. And as you heard, one of the things I ask people to do, get your hand and go like this and just feel how squishy you are, right? People are squishy, but that's how hard the plastic dishes are that we grow cells on. And cells don't like that. There's nothing in your bodies anywhere near as close to that stiffness. So cells don't behave when they're on plastic dishes, but we learned a lot. We did those experiments and we actually got these cells from umbilical cords. We go to the hospital, babies will be born, we get the umbilical cord that has blood vessels in it, and we take out the endothelial cells and grow them. So it was useful. We learned quite a lot from that. And then by 2002, gone gray, got old. What we've learned in that time was how to take these cells and put them in a biogel, so a bit of a squishy gel that's about the stiffness of your body. Here, the cells are much happier, and as we saw earlier, they will now make these little blood vessels, and these are about the size of the capillaries. So that was cool, and again, we learned a lot about blood vessels from these kind of assays. And one of the things that we wanted to do, we took our mouse, because I said they can be useful, and we asked, are these really blood vessels? Can they really work as blood vessels? So we put these human blood vessels in a mouse, and they are blue, and the mouse vessels are red. And what you can see here is that the human vessels hook up with the mouse vessels. So we got mouse blood flowing through human blood vessels in a mouse. And again, super cool. Did a lot of work on that, published some nice papers, learned some good things. This was done with my good buddy, Steve George, who used to be here, is now at Davis. And this is where my bioengineering really began. So I worked with Steve on these mouse projects. And then one day we're sitting in his office and he says to me, I don't know about you, I'm kind of bored with what we're doing. I'm like, yeah, me too. I feel like we've gone as far as we can. And I said, well, he said, what would be a really cool transformative thing that we could do? And I said, well, look, we're making blood vessels in culture. Well, wouldn't it be cool if we could run blood through them and actually have tissues with blood vessels growing in a dish? And so we thought, okay, how would that work? And so we literally sketched it out on a yellow pad on his desk. And doesn't make much sense to you here, but it will. But that was the first drawing of how we might blend engineering and biology. And it became this thing. And you could think of the red one as the artery, the blue one as the vein, and lots of little capillaries joining them. And we got a grant from NIH, and we actually got this working. So it wasn't great, but it was a proof of concept. I can't sing the Jeopardy music, but you can do it yourself. So time passing, about 15 years later, did a lot of stuff in the interim, but we got this working really nicely. And so what you're seeing here is a channel that we've created. We've engineered this channel and this one, and they represent the artery and the vein. And then we grow human capillaries, these little blood vessels, these little human blood vessels, and we connect the artery to the vein, just as it is in the body. And then we've run through this red dye, because it looks like blood, and that runs through the vessels and then comes out the other side. So we can run blood through vessels in a dish in the lab, which is kind of cool. And we can put in these fluorescent microspheres, so you can see those flowing through. And then, of course, we put in blood. And so there are areas of low flow and areas of accelerated flow here. So that's our vessels and our little platform. And this, the cuticle on your nail, you put a microscope on there and you can see little blood vessels. And again, you can see areas of low flow and high flow through here. So it looks very much similar to what you see in the body. So we've really now created blood vessels in a dish that carry blood. So we did what we wanted to do. And if you're interested in what this looks like, there's 3 layers. There's this plate. This is where all the goodies are, the microfluidics and the transparent bottom. And I'm going to pass this around if anyone wants to look at it. This is one of our plates. And if you hold it up, you can see this chamber here. This is where I filled it with blue dye. So you can see these little chambers. There's 12 of them on every plate. And these lines here are the artery and the vein. So we've now got 12 little tissues with blood substitute flowing through on one of these plates. So that's nice. So what are we going to do with it? So there you can see the blood vessels in that chamber there. Couple of other pictures show you what it looks like. We can see this because the cells, the endothelial cells, we put a protein in that glows in the dark. So you hit it with a laser and it glows green. It's called green fluorescent protein. Very exciting. And what's cool is it's originally from jellyfish. So glow in the dark jellyfish. Somebody took that and put it into cells so they glow green, which is kind of neat. Here they've got a red protein, and you can see this is the artery. These are the little capillaries coming out of it. So again, that's what we want. And if you're wondering how the blood flows, it's a hydrostatic head. So water finds its own level. We put our blood substitute here, and less of it there, and then it flows down the channels until these things even out. So all very simple. So we've got this model. So now we can model vascular diseases. We can create a vascular disease on this.
Chris,
So
how long can that flow happen till you have to read?
one version of our platform, probably about 36 hours. Another version, 12. It varies. So port wine birthmark. So this is, as I said, a vascular disorder. It starts off with this sort of blush on the skin, but over time it grows and gets darker, becomes like this. If any of you don't like looking at kind of gross things, just close your eyes and I'll tell you when to open them again, because this disease can get a lot worse. So here we go. You can see it can grow into these really large nodules. It's incredibly severe. It's difficult for these patients, as you can imagine. So, okay, it's gone. You can open your eyes again. So it's a nasty disease. When it spreads to the brain, that's when it's called Sturge-Weber syndrome. So that's what we're working on. And this is a disease caused by abnormal capillaries. So again, here's our normal artery, nice little capillaries. But in port wine birthmark, they get big and twisted and dilated and ugly. So you can see this is the PWB, and because they're all red and filled with blood, the surface of the skin looks very red. So that's the port wine birthmark. So my good colleague, Kristen Kelly, she's a physician here. She treats patients and developed, a colleague of mine, a way of looking at living blood vessels in the skin. And so these are the normal ones. They all look good. But in these port wine birthmark lesions, you can see these vessels are much bigger. They're much more dilated. And what Kristen does, she uses a laser and she'll go over and kill all of these blood vessels. And that helps the patients. But unfortunately, these bad vessels do grow back. So what we'd like is to have some drug treatment for this. So why do these vessels do it? Well, simply put, there's a mutation in the gene GNAQ, which causes PWB. So what does that mean? If you're not a hardcore molecular biologist, that may not make a lot of sense to you. So let's explain. Your body is made up of trillions of cells, you know that. And cells have a nucleus, this center part of the cell. And in that nucleus are your chromosomes. Your chromosomes are made of DNA. And DNA is a series of 4 letters repeated over and over. And it forms a code. That's your genetic code. And the sequence of these determines who you are. It will say whether you have brown hair or whether you have black hair, whether you have brown eyes or blue eyes, whether you're tall or short, whether you are susceptible to diabetes, whether you're more or less likely to get cancer. Everything about you is encoded in your DNA through this sequence. And that sequence tells the cell how to behave. And what happens is this gets translated into proteins. So proteins, when you look at me, hair, that's protein. Skin, lots of proteins. Everything in your body is proteins and lipids, but the proteins are the ones that make everything happen, okay? And that's coded in your DNA. All right, so what happens? Sometimes you get a mutation. One of these letters changes, for example, or a bit gets deleted. And what then happens is that this protein now has a mutation. The protein is different. It might not work the same. And that's what happens with GNAQ. So this is the GNAQ protein. This is the structure of it. So you can see what it looks like. So what does it do? So we're now in an endothelial cell, the cells that make blood vessels. This is the surface outside, the membrane that defines the inside and outside of the cell. And there are signals that say, dear endothelial cell, please make a blood vessel. And that gets transmitted across this membrane. And grossly simplifying, it causes this inactive GNAQ to become active GNAQ. And then that triggers a whole sequence of events, at which point the endothelial cells now make a blood vessel. So that's all well and good. When you've got your blood vessels made, this signal turns off, and this active GNAQ goes back to its inactive form, and the endothelial cells stop making blood vessels. All is good. But what happens if you have a mutation in this protein? What happens is, even though you get an off signal here, the GNAQ cannot go back to its inactive form. It gets stuck in its active form. And so the endothelial cells don't stop. They just keep making blood vessels. The vessels get bigger, and that's port wine birthmark. So we know the gene, it's a problem with GNAQ. So we want to make a model of that so we can test drugs and understand it. So to do that, we use stem cells. And you know that you start off as a single cell, right? The egg, you become a bunch of cells, eventually you become a person. That process involves stem cells. And these stem cells do what we call differentiate. They turn into all the cells of your body. Some will become neurons in your brain. Some could become gut cells. Some could be immune cells. And some will become the endothelial cells that make blood vessels. So we can take this process and use it. So we take a stem cell and we modify its DNA. So what we do, we put that mutation in. So that mutation that causes port wine birthmark, we put that into the cell. So now we have a normal cell and a mutant cell, and then we make endothelial cells from them. So now we've got normal endothelial cells, and we've got these mutant or PWB endothelial cells, and now we ask them to make blood vessels. And so what happens, here is our platform again, and you remember you have an artery, a vein, and the capillaries, the artery and vein and capillaries. The blood vessels are red. Here we're putting through green blood just for fun. So that green is the blood flowing through. And this is the development of these blood vessels over time with normal endothelial cells. So then the question is, what happens if we've got this mutation in the endothelial cells? Well, they form these big, nasty looking vessels, just as we hoped they would. And if you remember, when we looked at vessels in the patients, this is the unaffected control. This is the PWB with these big, nasty looking vessels. And this is our model, big, nasty looking vessels. So we've created the disease on a chip, as it were, in that platform that you're passing around. And so what we're doing now, we're adding drugs to this to see if we can reverse this and make it go away. Good news is we have 2 hits. We have 2 drugs right now that actually make this go away. So we're going to work with Dr. Kelly. What we want to do is formulate that into a cream, get FDA approval, then start putting it on patients and see if it'll make their lesions go away. So kind of fun and exciting. So the last couple of minutes, just going to touch briefly on this other disease, HHT. I've been working on this for a long time. I work with a disease charity called Cure HHT that helps patients with this disease. And again, it's a vascular overgrowth problem. Here you can see this big, nasty tangle of vessels in somebody's brain. Also happens in the lung and the gut. And these vessels are fragile and they can burst. And so the patients lose a lot of blood. They're anemic. Sometimes they get nosebleeds. You've had a nosebleed and you thought, wow, that's a lot of blood. And you lost maybe a mil of blood, right? Little, like, what's a mil? What's a mil in imperial? Very little.
Pencil eraser.
Pencil eraser of blood. A couple of pencil erasers of blood. And it feels like a lot, right? These patients can lose half a pint of blood gushing out their nose. It's traumatic, as you can imagine. It makes them very unhappy and very sick. So that's the disease. And we have recreated this on our chip as well. So again, these are fairly normal looking vessels developing here, but later on we're forming these big vessels. They're called arteriovenous malformations. And again, we've managed to recreate this on the chip. We've tested drugs. This one is called pazopanib. And just to walk you through this, this is the one graph I have. What we've measured is the lesion score. And down here, this is the control, and the gray is the lesion when we make the mutation. So you can see there's the control, and that's with the mutation. We've got lots of lesions. And then when you add the drug, you look at this last one, the drug makes the lesions go away. So the control, and this is the mutant with drug, and we've basically made the lesion go away. And that's very cool because a few years ago, I was involved in a clinical trial for HHT. It was done by Cure HHT, this charity. We got somebody from Dennis Sprecher from a drug company, gave us the drug. There's me. I was the scientific advisor on this clinical trial, and we used this drug pazopanib. And while it didn't work on all patients, some patients came back and said, oh my God, this has changed my life. I haven't had a nosebleed in weeks. I feel great. I'm not anemic. So we don't know why it only works on some patients, but not others. But it's just super exciting for us and for the patients that we now have a drug, and Cure HHT is developing other drugs as well. We're testing those in our platform. So yeah, it was just one of the most exciting parts of my career to see that happen. And so going back to my daughter, did you cure anyone? Kinda. We didn't cure them, but we did make them better. And so I'm kind of proud of that. And that's what translational medicine is about. It's taking basic research and turning that towards the clinic, trying to develop drugs and therapies that help patients. So, summarize body on chip technology, which is what we've been talking about here, stem cells, you put those together, you've got an incredibly powerful platform for recreating human disease in the lab in ways that you can test drugs and hopefully identify drugs that are going to help patients. And I've shown you a couple of examples where we're heading in that direction. We've also got a huge project going on cancer, looking for new therapies to treat cancer as well, using the same platform. And the most important slide always is the people that actually do the work in the lab. And I see some young people in here who I know will be on these kind of pictures in other people's labs. And the people I want to call out specifically, Duke Fan here is this brilliant bioengineer. He did a lot of the work getting this platform working. Jen Fan here, she did the HHT work. She now has her own lab at Tulane. This is Will Van Trite. He's just graduated. He did all the port wine birthmark work. And then obviously collaborators, Kristen Kelly's been just fantastic collaborator on the port wine study. My long-term collaborator on all things bioengineering, Steve George. Always have to thank NIH, your tax dollars at work, which pay for the work that we do. The Sturge Weber Foundation that supports people with port wine birthmark, Sturge Weber. Cure HHT that helps HHT patients. And of course, for all their support and for being here, the Sue and Bill Gross Stem Cell Research Center. And so with that, I'd be happy to take questions. Thank you very much.
So Chris, before, 1 second. So we don't have, UCI Media, who used to record our lectures, is no longer here. So we're using this gizmo called the Owl. So I'm not going to be coming around with a microphone tonight, if any of you are familiar with that. Shout out your question. I'll come to you if you need to. Chris, can you please repeat it from here so that,
yes.
Perfect.
Please.
Okay, I'd like to know how the drug pasinaviv, how does it work? Does it selectively dry up certain capillaries? Does it cut off some of them? Does it make them dry up? Does it make them atrophy? Does it make them occlude? What happens?
Good question. So the question is, how does pazopanib work? What does it actually do to the blood vessels? So there's a key pathway in endothelial cells that drives them to make blood vessels and helps maintain blood vessels. It's called the VEGF pathway, VEGF. And VEGF is a growth factor that floats around. That's one of those signals that says make a blood vessel. And it goes through a receptor on the surface called VEGFR2, and pazopanib blocks that signal. So the endothelial cells lose the signal to make a blood vessel, so they stop making blood vessels, which is good. It can also cause them to regress, we think. It's a little more complicated than that in ways we are still trying to figure out. It's not immediately apparent to those that work on HHT why the drug should work as well as it does. So partly it's by destabilizing vessels, partly it's stopping vessels grow. We think it might also help with the bleeding directly by when you take it away, the vessels stabilize. So when you've got that signal there, the blood vessels are all excited, they're all twitchy and ready to go. When vessels are like that, they tend to be leaky. But if you can calm them down, get them to sort of chill out a bit, they become less leaky and less fragile, which may be why there's less bleeding. But it's a great question. It's something that people in the field are still working on. We actually have a project on that, and we have some leads actually as to why the blood vessels are fragile. and have some ideas about how we can make them more robust. So yeah, good question. Thank you.
How do you get the pharmaceuticals interested in developing drugs for these two diseases?
Question is, how do you get pharmaceutical companies interested in making drugs for rare diseases? By and large, you don't. The problem is in the very description. They're rare diseases, so there's no market. So pharma companies like nothing more than having a drug that everyone can take forever, right? They don't like cures, right? Cures aren't good because your market goes away. So a drug you take every day, they love that, and they like lots of people doing it. So rare disease, there's no market. So if they do develop a drug and spend billions of dollars doing it, they want their money back. So the drug's going to be just super expensive. So what we do, by and large, and I will just say there are a couple of companies now that are starting to work on rare diseases, which is great, but there aren't many. The big ones tend not to. So what we do is what's called drug repurposing. So you take a drug that is approved and in the clinic for treating one disease and use it to treat another. So pazopanib at tenfold higher doses is used for cancer patients. But we found that much smaller doses are helpful for HHT patients. The two drugs that we've identified for port wine birthmark are already approved drugs for other diseases, but the same pathways turn out to have a role in port wine. So we think those drugs are going to be effective there. So drug repurposing is mostly what we do because it's cheaper, right? You don't have to develop the drug. You just got to show that it's safe in this indication.
So before we go to the next question, can I just insert, Chris, can you comment? Because what a beautiful talk, but I think really bring home the connection between being able to generate a platform like this that uses human cells to be able to do screening for effective drug repurposing.
Yeah, that's exactly right. You said it beautifully. If you want to repurpose drugs, you've got to have a platform to figure out if they're even going to work in your disease. So by doing what we do and generating these disease models, and they're in a dish, so it's easy to do experiments, we can screen a lot of different drugs and find out what works. So we're doing that in a couple of rare diseases. We're also doing it in cancer. So it turns out that a drug that might be approved for one cancer hasn't been tested in another. And so we can test multiple drugs on multiple different tumors and find out maybe this thing that was approved for lung cancer actually works for this rare kidney cancer. So it's all about repurposing and having the right platforms to be able to do that. As that lady there, then that gentleman, then this gentleman.
Did I hear you say that it was a cream that was applied for the HHT? Or how is?
No, the HH, so the question is, what's the formulation of the drugs? The HHT drug, pazopanib, is a pill. There's another drug that actually is injected. For port wine, what we would like to do is formulate the drug in a cream. So because it's a surface lesion, right? It's on the skin, having a cream would be great for that. So that's what we would like to do there. And that gentleman.
Have you tried, or is there anybody else using your platform to develop new drugs?
Question is there anyone else using our platform? Yes, there are other academic labs using it. And what my company does is drug screening for pharma. So the company has worked with 7 of the world's top 10 pharma companies to test drugs in these kind of tissues, mostly in the cancer space, as it turns out. So yes, platform is being used for drug discovery and validation. Yeah.
So what is the angiogenesis approach you're using for cancer? Not just the drugs, but what kind of how are you manipulating that?
Yeah, great. That's a nice question. Question is, how are we manipulating angiogenesis in our cancer studies? So we've just published well, I have to say, it's a very cool study. This was really driven by a postdoc in my lab. It's quite brilliant, Stephanie Hashi. And so what she did was set up a breast cancer model, a triple negative breast cancer, which is very aggressive breast cancer, in our platform, and we added a drug called paclitaxel. And it sort of worked, but there were two issues. One, it's quite toxic and it damaged the blood vessels, and it also killed the tumor a little bit. So what she did was decided, let's make the blood vessels healthier. So she put in another drug that helps to maintain the blood vessels. And what that meant is because the blood vessels are better, they carry more blood, they carry more drug into the tumor. When she did that, the paclitaxel worked way better at killing the tumor because now we're getting more in. So by manipulating the blood vessels, you can actually increase delivery of drug into the tumor. The other thing you can do is put in drugs that disrupt the blood vessels. And what that does is deny the tumor nutrients. So that slows the tumor down. So you can go either way by manipulating blood vessels.
Okay, thank you.
Any more questions? Yes, at the back.
Could you explain about the dish? I look at it, but I don't know what to add. So I look at what is it. You basically just platform, when you put the stem cell inside your flow and.
Yes. Let me go back. Where's my pre-picture?
When you look at the dish,
Yeah, so what you're seeing here, this is the plates that I handed around. And it's got these, each of these is an individual tissue, and it looks like this. So what you're seeing that's blue is this area here, which is this area here, right? And what's in there is the tissue. So this line here is the artery. So you can see this is the artery here. This is the vein. So the flow comes in here along the artery, across the capillaries, and then out the vein. So that's what you're seeing. So all these pictures of blood vessels that you're seeing, they are all in this little chamber here.
So the flow would be.
So the flow goes from here, it goes to there, and it goes from here to there. And this is a higher pressure side than this. So what that does, it forces blood from the artery across these blood vessels to the veins. So that's how we drive the flow.
So just to interject there, was that part of your engineering design phase is getting those pressures right? So that the
Yeah,
vessels
so
actually-
I haven't put up the pictures here, pretty as they are, but we do a lot of computational fluid dynamics. So we take pictures of these, we import them into this engineering program, and then we simulate the flow and the pressure through the vessels. And by doing that, we can dial in the flow rate that we want. So we say, we want a flow of, say, 2 millimeters per second, and the program will tell us how much blood substitute to put in these wells to generate that hydrostatic head. So if your head's like this, you've got high pressure, high flow. And if it's like that, you've got lower pressure, lower flow. And so we can manipulate the flow rates that way.
And does that affect capillary development? and
Oh, it
maturation?
does. So for the so hello, Al.
Sorry.
The question is how the flow rates affect the vasculature. So it turns out with the HHT model, if you have low flow through there, you get a lot of vascular overgrowth, but you don't get those big vessels. If you increase the flow, now you get the big vessels. So the formation of the big arteriovenous malformations is dependent on high flow and high shear. And that's been reproduced in zebrafish, it's been reproduced in mouse. So that's a thing. You have to have the high flow to get the big lesions. Yes, sir.
Can the capillaries that were grown, can they handle the same pressure as in the body, for example?
I don't, well, so the pressure at the level of capillaries is quite low.
Sorry, what was the question?
Oh, I'm sorry. Can our vessels handle the pressure that you see in the body? Yes and no. We model the very tiniest vessels, which are the capillaries, and yes, we can handle the flow rates and pressure you see in the body. The pressure and flow you get in arteries is way higher than what we see or even try to model. So we're very specifically focused on the smallest vessels as opposed to the really large ones. Yes.
You guys ever tried to scale up?
Have we ever tried to scale up? Yes. We have a model actually of an artery. It's a lot simpler than this. And there we do use a pump to pump fluid through under high pressure. And we developed that to try and model atherosclerosis, which is very much a high flow artery disease. Even there, for the engineers among you, we can't get the Reynolds number high enough to generate turbulent flow. So it turns out it's not optimal for atherosclerosis. Some aspects, but not all. The other question that gets asked all the time, and I'm surprised it hasn't come yet, can we take these blood vessels and put them back in the body? Which is obviously where we started with this, making blood vessels and grafting them into mice. And I've worked with people to try and pre-vascularize tissues. So if you're making a tissue for transplantation, you would like it to already have the blood vessels. So when you put it in the body, you hook up the blood vessels and it instantly gets fed. Otherwise, it's going to die. It turns out that we're really good at making these small vessels, but in trying to, in the lab, make small vessels hooked up to big vessels is beyond us or anyone right now. So we can make vascularized tissues and transplant them into mice, and they work. That's what I showed you earlier, where that blue and red vessel hooked up. But that's a tissue about the size of my thumbnail, probably a millimeter thick. As soon as you try and make tissues that are bigger than that, might actually be useful at our human scale, it doesn't work. So, yeah. So somebody will solve it one day. I'll be retired by then. It's a tough problem. Yes, I think it's a gentleman there and this lady here.
Yeah, so for this model, and you only put in one cell type, or you have other?
Oh, how many cell types are in there? For the ones you've been seeing, there are 3 cell types. There's endothelial cells, there are fibroblasts, and there are pericytes. Pericytes are cells that wrap around the blood vessels. I skipped over that. We've got a brain model, and there we've got endothelial cells, fibroblasts, pericytes, astrocytes, and neurons. So 5 different cell types. We're about to put microglia in as well. So we'd have 6. So basically creating a vascularized mini brain. So we're going to study Alzheimer's and Huntington's disease with that. Lady down here.
Yeah, for the port 1 birthmark studies with the genetic mutation, is that mutation in all cells in the body? Because it looks like the birthmark is very localized.
Yeah,
other tissues also.
Yeah, very insightful question. No, it's not. It's what's called a somatic mutation. So there are some mutations that you're born with. So those are congenital. And there are others that appear later in life. Those are called somatic. And they happen just in one cell, usually, and then the cell makes copies of itself and it spreads. So no, it just happens in some blood vessels where this GNAQ gene is, but not all blood vessels. So, and it's very interesting that it's mostly on the face and it's mostly unilateral. The lesion doesn't spread across the midline. It tends to stop halfway. And that tells you something about development, basically. But no, it's a good question. It's not everywhere. It's very localized just to those blood vessels. And the fact that it's all the blood vessels in one zone suggests that the somatic mutation happened very early during fetal development. But yeah, nice question. Yes.
What basic mechanism accounted for the difference or dependency on the flow that now impacted how the vessels behave?
Right. So why does flow influence how the vessels behave? It's because of the shear. So shear is basically the friction of fluid flowing over a surface. And if you think of a stream with like grass or the seaweed in the sea, as the water moves, that it sort of flows back and forth. So there are proteins on the surface of the endothelial cells that do that under flow. They sense the flow, and that tells the endothelial cell where it is. So if it's in an artery, there's a lot of flow. So the endothelial cell goes, oh, I'm an artery, so I better behave like an artery endothelial cell and do what I'm supposed to do. If it's just kind of gently going back and forwards, the endothelial cell will be like, I must be in a capillary. It's just gentle flow back and forth. So I'll do what capillary endothelial cells do. So the cells sense the flow and it kind of tells them how to behave. We can reproduce that, which is nice.
Is that useful for the scaling up for larger?
It makes it difficult because the amount of flow and the shear you have to generate, your heart is a very powerful and efficient pump. And so try and recreate that in the lab is not trivial. So we don't do it. Yes.
Your brain model.
Yeah.
That can you or do you think that in the near future that can you use that brain model and do the test for like BBB penetrance of the like chemo medications or like the study for the like preventing the metastasis through the like bloodstream.
Right. So the question is, can we use our brain model to look at the blood-brain barrier, blood-brain barrier function? Yes, absolutely we can. And for me, it's coming full circle. My PhD project was working on blood-brain barrier. So it's kind of cool to come back to it. So we have a really good blood-brain barrier. And we did this experiment, which I love. We took our regular vessels and we put a tumor in there. And then we ran a drug through the blood vessels. It got out and killed the tumor. So then we repeated that in our brain model, at our brain vessels, and put the tumor in, ran the drug through. Tumor didn't mind at all because the drug couldn't get out of the vessel because of the blood-brain barrier. So for those of you who don't know, the blood vessels in the brain are very different to blood vessels elsewhere. Most of your blood vessels have a certain amount of leakiness, but the ones in your brain, because the brain is so delicate and sensitive, the endothelial cells there are very strong and strict gatekeepers. They don't let much get in. They control what gets in, and they really don't like drugs going in, those kind of molecules. So they pump them back out. And our model does the same. So we can use it to identify drugs that can get in and stay in. And that's something we're super excited about doing. And for metastasis, yes, it's a very good model for that. We have done those studies. So for example, we can set up a vasculature and then we can put tumor cells into the input and they go through, they get stuck in the vessel, then they get out and form a tumor. And I don't have it here. I've got this amazing movie of a tumor growing and then becoming what we call going through an epithelial mesenchymal transition. And that's when the cells get into the blood vessels and leave the tissue. So we can see both ends of metastasis, the seeding or the seeding of the blood, and then the exiting of the blood into a tissue to form a metastasis. So yeah, those can be and have been studied in the platform. Thank you. Yes.
Okay, last question.
Yeah, what do you comment on using autologous versus allogeneic cell or organ models?
Yes.
For
Question is, yeah, can I comment on autologous versus allogeneic models? What that means is an autologous model means all the cells came from the same individual. An allogeneic model is when the cells come from different individuals. Most of what we do, it doesn't matter. So our model is allogeneic. So the endothelial cells, the stromal cells, all from different donors. Where it's important is when the immune system comes in, because the immune system, what it does, it recognizes not you. So if there's anything in you that's not you, the immune system will destroy it. That's how it works. So if you do a transplant, put somebody else's heart in, your immune system goes, that doesn't belong to me, and tries to kill it, which is why you have to take drugs. So in our platform, we have done studies where we want to, for example, have immune cells killing tumor cells. There you need to have them autologous. So in that situation, what we do, we get tumor biopsies from patients, we take blood, and then we've got their immune cells and their tumor. We can put those together in the platform. So at least that part of it is autologous. The rest is still allogeneic. The beauty of iPSCs, stem cells, is that you have the possibility of making all the cells you want from the same source. So you can take a single iPSC, make the blood vessels, make the tumor, maybe make the immune cells. That's not possible right now, but in theory, one could do that and make a fully autologous system. Thank you.
Wow. So I just want to say thank you, Chris, for one of the best, I think, most clear lectures that we have ever had. That was fantastic. And as you can see, thanks to the audience for just some phenomenal questions and really staying with us through this journey. So thank you, everyone. Please help me thank those speakers.
Thank you.